Smoking is a cause of several serious and fatal conditions, including lung cancer, heart disease, and chronic obstructive pulmonary disease. In our laboratories, we're working to understand exactly why tobacco smoke is so harmful and to see if there are ways in which we may be able to reduce the potential to cause harm. Our research broadly includes three main areas of science: chemistry, biology, and technology. First, we'll look at chemistry.
Tobacco and tobacco smoke are really quite complex. There are thousands of chemicals in tobacco smoke, and the first thing we have to do is to collect the smoke from the product. So we smoke cigarettes on smoking machines, and they are smoked to a number of different smoking regimes. One that's part of an international standardized method, and also to more intense smoking regimes, which are believed to represent more closely the way in which people smoke. Cigarette smoke is an aerosol, so it comprises particles and gases and vapours. We have to collect those. Generally, we collect them separately. We collect the particles on a pad, and that constitutes most of the tar in cigarette smoke, and we collect the gases and vapours separately. We extract the tar and chemicals that are collected on the filter pad into a solution, and then we would analyse that extract via a range of techniques. Most of them are chromatography based techniques, some what's called a gas chromatography for the more easily separated chemicals in the tar, and others that are less easily separated and are larger molecules, we have to use a technique called liquid chromatography. The particles are in the size range of 100 to about 250 nanometers. So that's between 200 and 300 times smaller than the aerosol particles that come out of a deodorant spray. We use a variety of techniques to measure the size of the particles in tobacco smoke. These are very similar to the techniques that are used for measuring the particles in vehicle exhaust. We need to understand the size of the particles because that can influence where and how the particles are deposited in the lung. And that's important because the chemicals in the particles can be absorbed into the body in different ways and at different speeds.
In the lab, we're also conducting biological research that includes, for example, researching new cigarette exposure systems and models of tobacco-related disease. These models, which consist of human cells that can be exposed to tobacco smoke, could help us to understand the development of tobacco-related disease. In the future, such models may also act as screening tools for assessing cigarettes that have been modified to produce less toxicants in smoke. The work shown here is examining how exposure to smoke may affect the body's ability to repair damaged cells. Some tobacco products, like snus, are described as smokeless because they're designed to be held in the mouth rather than burned. With no smoke to collect and expose cells to, British American Tobacco has developed the snus engine. This mimics the action of the mouth, allowing us to collect the chemicals and compounds that would be released into the body. These can then be analysed using similar techniques to those used for smoke analysis. This kind of research is very important as it contributes to an understanding of how tobacco-related diseases are caused and helps us consider ways in which the products could be modified to reduce the possibility of disease.
There are three major diseases associated with cigarette smoking: cancer, cardiovascular disease and chronic obstructive pulmonary disease. And we need to understand how these diseases develop. So we're using cultured cells in the laboratory to try and see how these processes work. These diseases are not just associated with tobacco, and when we present these models amongst the wider scientific community at academic conferences, they do create quite a lot of interest.
Developing technologies to modify tobacco products or treat tobacco is the third area of research.
Cigarette smoke comes from burning tobacco, and tobacco is a plant, And so we need to understand the processes that are taking place within the plant cells. And our laboratories in Cambridge are trying to understand the metabolic processes within the plant cells and to see whether they can influence the levels of the precursors in the cells in the tobacco plant to the smoke constituents.
Snus provides a good example of the potential for using technologies to try and address the hazards of tobacco use. By treating the finely cut tobacco in a process similar to pasteurisation, we can reduce the levels of chemicals called nitrosamines that are linked to the development of cancer. We're working hard to understand tobacco and the mechanisms by which it causes harm. There's still a long way to go, but we believe that we're on the right track.